Thank you. Pleasure to be back to talk to you once again. Yes, Fiat Lux, let there be light. Fiat Lux IV, don't worry, you don't need to have seen the other 3 episodes. This particular one was added after the original trilogy, so I'm still calling it a trilogy. It's just a trilogy in 4 parts, okay? And this particular episode, episode 4, is Colour in the Cosmos. So I'm going to be talking about, before we get stuck into the cosmos, I'm going to be talking about how we as humans perceive colour, because it doesn't make sense to talk about the colour of the stars or the colour of nebulae until we understand how we perceive colour. So it's perception followed by how we actually quantify colour before I get stuck into talking about the colours of stars and the colours of nebulae. I'm probably going to skip over something I might have added, but it would probably make the talk too long, some aspect of spectroscopy. I'll be saying a little bit about going beyond colour, and then I'll be summarising at the end. Could I just ask for a quick show of hands? Is anybody here colourblind? Nobody. Okay, I won't have to exclude any of the audience then. That's great. Okay, so we start with the obvious question. What is colour? Is colour real, or is it just a pigment of your imagination? Yes, yeah. PowerPoint always tried to change the spelling of that back to figment, but there we go. So why do we have colour vision in the first place? A beautiful rainbow. Think what that would look like if we didn't have colour vision. It would be a fairly boring sort of thing. It is widely believed that we have colour vision. One of the main reasons we have colour vision is to make it easier to identify what it is we're eating. If we are, for instance, eating fruit, then we really need to know whether it's ripe or not. Simply because if it's on the next tree, we need to know whether it's worth walking to the next tree before we pick the fruit. So looking at that, 3 tomatoes, which of them are ripe and which of them aren't ripe? I presume you're all having a bit of difficulty trying to work out what's going on. But if you have colour vision, it is pretty obvious which ones are ripe and which ones aren't. So that's the conventional view. I think perhaps we have evolved colour vision simply to make it easier to read the underground maps. Because without colour, it's very difficult to see what's going on. As soon as you put colour into the mix, then everything falls into place. Okay. How do we perceive colour? Excuse me if you know this, but we just go through the basics. The human eye, light goes through the lens, hits the retina, and the retina contains a whole load of photoreceptors. There's a mixture of different photoreceptors. Some of them are colour sensitive and some of them are not. So let's just label them as 3 sets of colour receptors, those that are receptive to red and green and blue light. And the signals from those colour photoreceptors gets sent via the optic nerve to the brain. That's a rather simplified view of how vision works. We could ask, well, is that sort of the same as a camera? Because a camera has photosensitive receptors, pixels if you prefer, and the information is then sent to a computer for interpretation. So every digital camera will have a chip of some kind. Generally speaking, if we're talking about a colour chip, the chip itself is not colour. The chip is a monochrome chip, but it has colour filters on the top. So every pixel is sensitive to the same light hitting it, but every pixel has either got a red or a green or a blue filter manufactured onto the top. And if we look at what light is passed through each of these red, green, blue filters, I've chosen the camera that I was using at the time. These cameras, this particular response curve is unmodified. So this is not a camera that's been modified for astrophotography. This is just a normal daylight color camera. And you can see that the blue filter lets through blue at end of the spectrum. There's the spectrum along the top and wavelengths, if you prefer, along the bottom. The red, not surprisingly, the red end of the spectrum, and green in the middle. So that's exactly what you would expect. And it's very easy to look at these curves and say, well, that's exactly what I would expect for a color camera. 3 filters that let through mainly red, mainly green, mainly blue. And you might assume that human eyes see effectively the same thing. But if you were to plot the response of the human eye, it looks rather different. Yes, you get a big peak here for the blue, but look at green and red, not quite what you would expect. You might expect each of them to cover 1/3 of the visible spectrum, but no, the red and the green actually overlap quite considerably. There's enough difference that you can tell the difference between red and green, but it's perhaps not what you would expect. So although we might naively label these red receptors, green receptors, and blue receptors in the human eye, you can see that, well, actually, if you look at the red receptor, its response is finite for red light and yellow light and green light and cyan light and blue. The red receptor actually fires for any light coming into your eye, regardless of the wavelength. It's just higher in the yellow. It's not even highest in the red. So biologists don't refer to these as red, green, blue color receptors, though I will continue to do so in this particular talk. Biologists actually refer to it as the 3 receptors that are responsive to long wavelengths, short wavelengths, and mid wavelengths. But of course, for us, it's easier to think of them as red, green, blue receptors. There's a little bit of gap between red, green, and blue. That's where the rods sit. If I take away the color vision and just remind you that's where the rods are, remember the rods are responsible for night vision, rather important for astronomers, and notice that the response of the rods disappears by the time you get to about yellow. That's why astronomers use red lights at night. You can have the red light as bright as you like to give you color vision so that you can read your star atlas or see where your feet are, but as soon as the red light goes off, the rods are completely insensitive to the red, and so your night vision is preserved despite the fact that you've been looking in color. If you try that with green or blue, you'll find your night vision is destroyed. Okay, so you could ask, well, okay, right, we have 3 sets of color receptors, and that's why we build cameras with 3 sets of color receptors to mimic the human eye. But actually, some animals have more. So humans have red, green, blue. I'll continue to call them that. But some animals have very interesting color receptors. Look at that. There's the visible spectrum again. So there is an animal that has color receptors that peak, and it has what, a dozen of them? One of them actually goes into the infrared, and actually quite a few of them go all the way into the ultraviolet. Would anybody take a guess as to which animal this is? Mantis shrimp. Yeah. Now, I'm not too sure. I assume that they have that many receptors because it makes it easier to find food. But this is not a biology lecture, so we're not going to go into that. But it's quite interesting that this idea of having 3, red, green, blue, just the 3, is something that's definitely there in primates, but you can't assume it's there in the rest of the animal kingdom. So why did we end up with 3? Why did we end up with 3 primary colors in the back of our eyes? Well, it all comes back, if you take the history, it comes back to the time when, well, when dinosaurs ruled the earth, we were just these little tiny furry mammals. We were nocturnal because we couldn't come out during the day because we get stomped on or eaten by dinosaurs. 65 million years ago, the dinosaurs were wiped out, and then these little furry animals came out during the day because now it was safe to do so. So when we were nocturnal, there wasn't much point in having night vision, which was color, because generally speaking, color receptors don't work too well at night. But now we're active during the day, we can start to develop color receptors. And so basically, 65 million years of genetic mutation takes us from a limited number of photoreceptors up to the 3 that we have now. I'm not implying that humans at the end here are the peak of evolution. One assumes that in the future we'll probably go further and we'll evolve into, well, I don't know, but probably something else. A reminder that we're talking about visible light because our eyes, of course, are sensitive to visible light, and the reason we are sensitive to visible light is because the transparency of the atmosphere is close to 100% for those particular wavelengths. So most of these wavelengths in the visible part of the spectrum reach the Earth, and therefore our eyes and our telescopes will point to the sky and look in the visible part of the spectrum because we happen to have a dip in that region. If we wanted to look at the universe with infrared or ultraviolet, well, they tend to get blocked by the Earth's atmosphere, and so we would put a spacecraft up above the atmosphere, such as Newton or Swift or the James Webb Space Telescope or Spitzer Herschel, etc., for the infrared. That doesn't stop us putting a telescope working in the visible above the atmosphere. It's nothing to do with the absorption of the atmosphere. It's purely because of the turbulence that would affect the clarity of the images, not the fact that any of the light would be absorbed. If we come back to this idea of having 3 sets of photoreceptors in the back of the eye, if we look at that in a little more detail, well, it's a lot more complicated than my very simple diagram of just red, green, blue dotted over the back of the eye. There's a whole set of photoreceptors in there. The cones and the rods are mixed together. We don't need to worry about exactly how. But one thing that is quite important is that there are a whole load of cells in there which aren't the photoreceptors themselves. They are neurons. And for those of you who think neurons are just brain cells, no, there are neurons in the back of your eye, and they are there to do image processing. In other words, the photoreceptors pick up the light, but then these neurons process that image even before the signals get to your brain. Those images are being processed by these neuron cells. So one way to think of it is to say, well, when we take a picture, we are picking up effectively red, green, blue from the individual pixels, each of which has got a filter, red, green, blue. And we are measuring how much red, how much green, how much blue is hitting the pixel, hitting the sensor. And that information is almost directly transferred into, right, there's your image. Okay, if you're generating a JPEG image, some of the colors will be played around with, but essentially the amount of red, green, blue that hits the sensor is the amount of red, green, blue that you see in the final image. That's how cameras work. That is not how the human eye works. It is rather different. The photoreceptors and the neurons are such that the photoreceptors will pick up the red, green, blue light. But even before it goes to the brain, those data will go through a neural network. Literally, the neurons are connected together such that information from the red, green, blue receptors doesn't get scrambled, they get combined in different ways, such that what actually gets sent to the brain, this is the neurons in the back of your eye before it goes to your brain, the neurons in the back of your eye effectively take things like, for instance, let's take the amount of red and the amount of green and subtract half of the amount of blue, and that is the information that gets sent to your brain. Not only that, but it's not only taking combinations of colors, it also takes combinations of what these photoreceptors are picking up and their neighbors, and maybe their next nearest neighbors as well. So it's much more complicated. It's not simply a pixel detects red, that's what you see in the image. When it's a question of human perception, things are very different. Let me convince you that what you think you see is not what you actually see. This is from a web page that's down here, so you can check that what I'm about to tell you is correct. I would like you to look at these 2 cubes, and if you look at this cube on the left-hand side, it's a sort of 5 by 5 by 5 Rubik cube. Just look at the top face, which has got 25 tiles on it. Can you count for me, please, the number of blue tiles on the top face of the left-hand cube? Have you all got a number? And can you do the same, please, for the yellow tiles on the right-hand cube? I heard a 6. Most people seem to say 4 blue on the left and 7 yellow on the right. Are you prepared to put money on that? You can tell from the tone of my voice that there aren't 4 blue on the left and 7 yellow on the right. You may be interested to know that the 4 blue tiles here and the 7 yellow tiles there are actually the same color. Yes, I'll say that again. Those tiles are actually the same color. Let me take away all the pixels other than the ones we are interested in. I'll take them away. Dun, 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 dun. That is what you were actually looking at. You weren't looking at 4 blue tiles on the left and 7 yellow tiles on the right. You were actually looking at gray tiles. And the neurons in the back of your eye were doing some image processing and sending signals to the brain that it thought that you wanted. The fact that these are a bit bluer than all the other pixels around them. The fact that those were a bit more yellow than all of the other pixels around them. But your eyes and brain in combination can easily be fooled. So when you think you're looking at something that is a particular color, beware. It is very easy to show that you don't necessarily see colors the way you think you do. And it's not just a question of colors. This idea of the neurons in the back of your eye processing information before it gets to your brain, that's true not only of color, it's also true of intensity of light. So that's not really what this talk is about, but let me just show you that it's true for non-colored objects as well. If we're interested in the square on which we have an A, and the square on which we've labeled a B, it's not the letter we're interested in, it's the square on which they sit, I can ask you which of those is the brighter of the two, which is the darker grey or which is the lighter grey. It's like asking you which is the brighter of these two stars. We think we can tell the difference of brightness. But when I show you what the actual brightness of these two squares is, let me not touch them, let me just mask out some of the other pixels in the image. Now, if you look at the square on which A sits, it is exactly the same shade as the grey mask on either side. And if you look at B, it is exactly the same shade of grey as the mask on either side. So the square A and the square B are exactly the same shade of grey. They're not similar, they are exactly the same. Now let me take those two masks away. Now you know that that is the same shade of grey as that. You know that's true because I've just shown you it. But the image processing in the back of your eye and in your brain almost refuse to allow you to see that. So you do have to be aware of the fact that if you are trying to judge the brightness of two things or the color of two things, you cannot believe your eyes. Hopefully, a camera records it with some fidelity, but human vision is subject to these sorts of, you might call them optical illusions. It's just the nature of human perception. So if that's the case, how do we quantify color? A spectrum. Oh, sorry, not that spectrum, no. We quantify colors usually by thinking about a spectrum. So good old Newton here trying to reproduce the album cover of Pink Floyd's Dark Side of the Moon. So we know that white light can be split up into colors. Fine, okay. And we know that whenever light passes through an optical instrument, whether it be a prism, a diffraction grating, a microscope, a telescope, etc., then the wave nature of light starts to manifest itself. And I'm sure you're aware of the fact that we can take the individual colors of the rainbow and we can assign them which particular wavelength of light corresponds to that perception of color that we see. 400 nanometers wavelength at the violet end of the spectrum to 700 nanometers wavelength at the red end of the spectrum. But a linear scale of colors is not sufficient for us to fully comprehend the nature of colors. Because, for instance, that's all the colors of the rainbow, but there's no brown in there, there's no beige in there. Yes, there's red, orange, yellow, green, blue, etc., but that is clearly not all the colors that can exist. And that's why when we're trying to quantify color, a simple linear scale is not good enough. And after lots of debate and lots of international discussion, it was agreed that one way of representing colors is in a two-dimensional space. And I am not going to worry too much about what these two axes are. I'm going to sort of fudge over that. One axis is, in a sense, how red is it compared to blue? And the other axis is sort of how green is it compared to blue? So we don't have to worry about exactly how it's calculated. All we have to care about is the fact that this is an international standard. So every country in the world agrees that this is one way of representing colors. And all colors can be quantified in a plot like this, a so-called color space. But one thing we absolutely have to be aware of is the fact that although spectral colors exist there on the outer part of that representation, when it comes to thinking about what colors we can represent in the middle of this particular color space, we have to be aware that every display, every device that's capable of producing an image, such as a TV or a data projector or my laptop, etc., they will not necessarily give you displays that look the same. Any particular digital display will be limited by producing primary colors, red, green, and blue, and it will be able to represent any color in that triangle where each corner of the triangle is primary red, primary green, primary blue. And so what I'm seeing on my laptop here is not quite what you're seeing on the screen, because the data projector and the laptop are two different digital devices. So any particular manufacturer will try and arrange it so that the red and the green and the blue are as far apart as possible to give you the largest triangle as possible, because you can generate any color by mixing red, green, and blue. You can generate any color inside that triangle, but you can't generate colors outside the triangle. Notice that all of these colors on the left-hand side, roughly where the green is written, all of them look pretty much the same. This projector cannot distinguish a lot of colors outside that triangle, and that is simply a limitation of digital technology. If the red and the green and the blue were further apart, you'd have a larger so-called gamut, a larger collection of colors. So that's something to bear in mind. You might generate an image, you give it to somebody else, they look at it on a different monitor or a different TV or a different iPad or a different projector. They will not necessarily see the same colors. Okay, so I've given you an introduction into what colors are and how humans perceive colors. So let's deal with the colors of stars. When we come back to this representation of all possible colors, we can ask ourselves, well, what colors of stars exist? If you plotted all the colors of the stars in the Milky Way, how would they be scattered over this particular diagram? Well, the answer is they wouldn't be scattered particularly, because every star has to obey the laws of physics. It's called blackbody radiation. If you take an object and heat it up, it radiates light, not arbitrary wavelengths. There's a very particular set of wavelengths that are emitted by an object of a given temperature. Again, we're not going to turn this into a physics lesson, but it's the so-called blackbody curve that tells you at a particular temperature you would expect these wavelengths to be emitted. And it means that the colors of stars must be somewhere on that curve. It looks rather limited, and you might think that there ought to be, well, are there no stars down here with this color or that color or this color or that color? Nope. Stars must be somewhere on that line. Cool stars like red giants on the right, and hot stars like A or B class stars on the left. You cannot have purple stars. You cannot have green stars. That might be a bit surprising, but the only colors of stars you can have are reddish or orangish or yellowish or whitish or bluish whitish. It never goes through green. It goes from red through white to blue, but not via green. So let's have a look at the actual distribution of colors that are coming off each of these stars of different temperatures. I mean, we can get an idea of what the colors are simply by heating something up. We don't have to go to stars. We can take a piece of metal and heat it up, and we realize that the relatively cool parts are going to be emitting a lot of infrared radiation. As it gets hotter, there's a hotter part of the bar. It's emitting a lot of red light and orange light. As it gets hotter, it tends to emit orange to yellow. And if it gets really hot, then it's what we call white hot. You would have to get metal very hot for it to start to go into the blue. But you can see that there's a definite gradation of colors there. So let's see what's actually happening. Here we have a little graphic in which we have a temperature scale up here. And on the left, we're seeing what wavelengths of light are emitted by an object of a given temperature. So here is wavelength, from short wavelengths out to long wavelengths. There, of course, is the visible part of the spectrum. So ultraviolet is to the left, shorter wavelengths than the visible rainbow here, and infrared is to the right. So most of what's in the right-hand side here is infrared radiation. And the curve here shows you how much light is coming off each of these wavelengths. at a temperature of, what are we at the moment? We are talking about a body which is 1200 Kelvin. And strictly speaking, it doesn't matter what that body is. It doesn't matter whether it's a lump of iron or a star. If they obey the same laws of physics, you get the same radiation coming off an object at the same temperature. So let's just bung the temperature up and see what happens. Note that that's where the maximum is. This particular object at 1200 Kelvin is emitting a lot at this particular wavelength, 2 microns. Let's bump the temperature up a bit. So now we've gone up to 1500 Kelvin. Note what happens. 2 things happen. The curve goes up, in other words, more light is emitted. The hotter the object, the more light is emitted. And the peak at which it's the maximum starts moving to the left, from long wavelength down to shorter wavelengths. Let's bung it up a little bit further. We're now at 2000 Kelvin or so. Notice that now we have a peak that's still in the infrared, and now we're starting to emit some light in the visible part of the spectrum. Let's go to 3000 Kelvin. Now this thermometer on the right-hand side says that 3000 Kelvin is the temperature of a light bulb. I don't know many light bulbs that actually reach 3000 Kelvin, so I think that's a slight mislabeling. But 3000 Kelvin is about the temperature of a red giant star. So what light is coming off a red giant star? Well, it peaks still in the infrared, but there's an awful lot of red light. You can see here, red is quite high, blue is relatively low. So most of the light coming off a red giant star is actually in the infrared. But within the visible part of the spectrum, there is far more red than there is blue. There's a little bit of blue coming off a red giant, but most of it is red. That's why red giants look red, because of what's going on here. If we bump the temperature up still further to the temperature of our sun, then now notice the peak is no longer in the infrared. The peak has now been pushed to shorter wavelengths into the visible part of the spectrum. And the amount of light coming off in the blue and the red, very similar. All wavelengths in the visible part of the spectrum are coming off the sun. So ever so slightly more blue than red, but you hardly notice that. The peak is actually somewhere in the yellow to green. But essentially, the sun is emitting at all visible wavelengths, which is why the sun looks essentially white. If we were to push that temperature up even further, then we would get to this particular case well over 10,000 Kelvin. Notice now the peak is now pushed well into the ultraviolet part of the spectrum. And if that's peaking in the ultraviolet, that means we have much more blue than we have red. So again, there's a lot of light coming off these stars. In a sense, it's emitting at all wavelengths, but there's definitely rather more blue than there is red. So these stars would be whitish, but definitely with a tinge of blue, which is why we call them a sort of bluish white. So to remind you, if we go from low temperature to high temperature, low temperature stars look red. When we go to the temperature of the sun, they start to look white. If we go even hotter, they're still white, but with a bluish tinge. At no point do we get a green star. It goes from red to white to bluish white. And that's why there's a limited number that I showed you in the color space in the previous diagram. We can always represent stars if we wish on a Hertzsprung-Russell diagram, and on the right-hand, the horizontal scale here, we have a temperature scale. Due to historical reasons, cold is on the right and hot is on the left, so it's sort of 3,000 down here and 30,000 up here, and we can remind ourselves that this color here is not so different from taking an object like a chunk of iron, heating it up, and we get relatively low temperatures here giving us a dull red, and the higher temperatures give us yellow or white, and eventually we come to a bluish white. But again, there is no sign of green in this scale of the temperatures of stars. I want to say more about what's going on in this Hertzsprung-Russell diagram, but I think this might be a good place to take a short break. Thank you. Thank you. Tea break over, so let's continue where we left off. I was telling you about the colors of stars and the fact that if we wish, we can plot the colors of stars on a Hertzsprung-Russell diagram. where the horizontal scale tells us the surface temperature, cool on the right and hot on the left. And as a reminder, that's where the sun is. Our sun is sort of fairly average. It's there with a luminosity of 1. That's not surprising because this scale is the luminosity relative to the luminosity of the sun. And so obviously it's not as bright as some and not as faint as others. And the surface temperature of the sun is sitting there at about 6,000 Kelvin. So don't take it too literally, the fact that it seems to be sitting there on the yellow. That doesn't mean the sun is yellow. Remember, it's emitting over all wavelengths. It means that there's no obvious tendency towards the red or obvious tendency towards the blue end of the spectrum. So at the top here we have supergiants, really large, massive stars. At the bottom we have a whole load of red dwarfs, relatively low luminosity and low size. So let's just think about the scales that are inherent in the Hertzsprung-Russell diagram. The top right are the really big supergiants, and the bottom left, I know there aren't that many stars down there, are relatively small. So there's a size variation from top right to bottom left. But also we know from studying not just a few, but many millions of stars, we know there's also a mass range. The stars in the top left are very massive, and the stars in the bottom right are very low mass. And we know from stellar evolution, for those that remember the ABC of stellar evolution, we know that large mass stars burn up their fuel very rapidly, and so they have relatively short lifetimes, whereas stars down at the low mass end of the spectrum, even though they've got less nuclear fuel to burn, because of the lower gravity and the lower mass, their fuel lasts longer. So short-lived stars at the top and long-lived stars at the bottom. So we can actually use color of stars to our advantage. It's not, oh, aren't they pretty? We can actually use the colors to actually tell us something about, for instance, a star cluster like this, which contains possibly hundreds of thousands, if not millions of stars, with a good enough resolution, in other words, with a nice space telescope like the Hubble, for instance, we can map out the brightness and the color of these stars, and we can plot them in a Hertzsprung-Russell diagram. And by plotting out all of these stars, we can actually find the age of the cluster. Because if we were to take all of those stars and plot them, most of the stars would fall on the main sequence, the diagonal there from top left to bottom right. Yes, there will be probably some white dwarfs in there, and there might be some supergiants up there, but a lot of the stars in a cluster will fall on that main sequence. From blue supergiants at the top, remember, very short lifetimes, to the red dwarfs on the bottom with very long lifetimes. So if we see, when we plot the stars out for any given cluster, if we plot the stars on the Hertzsprung-Russell diagram and we see the main sequence going all the way from the bottom right to the top left, we know it must be a fairly young cluster. Because remember, these stars don't live very long. And so if it wasn't a very young cluster, some of those stars would already have died off and they wouldn't be there. So we can judge the age of the cluster by seeing at what point the main sequence finishes. So if we get the main sequence going all the way to the top, the cluster must be very young. But if we find when we plot all the stars in this cluster that it only goes so far, by knowing the mass of a star in that position, which we can judge from looking at lots of other stars, we can say, well, this cluster must be, let's say, 20 million years old. Whereas if the main sequence stops at that point, then the cluster is older because fewer stars have survived. And we can basically get an age line by looking at where that main sequence finishes. If the main sequence goes all the way up to the sun, for instance, our sun has got a lifetime of about 10 billion years. Currently, it's about 5 billion years old, and there's about 5 billion years ahead of it. So a star like our sun would have a lifetime of about 10 billion years. So if we plot a cluster of stars and we find they go all the way up to the sun but no further, then we can deduce that this cluster is somewhere in the ballpark of 10 billion years old. So simply by plotting and seeing what we get in the main sequence, and that's only a question of taking the luminosity and the color, plot it up, see where it ends, that tells you the age. So it's a very powerful technique to use the color of stars to tell you about the evolution of a cluster. Some people with far too much time on their hands have said, what would happen if we took all the color of all the stars in all the galaxies and basically said, what is the average color of the universe? Now, some of us might have asked that question, but not many people have got the time to actually try and answer it. But some scientists have said, well, we've got a lunchtime coming up. Let's work out for 200,000 galaxies within a distance of a couple of billion light years or so. Let's look at their spectra and effectively add up all the spectra of all the stars in all of these galaxies to get an average light, as if you took the entire universe and just scrunched it up into a patch of sky and said, what color is it? So that's an interesting question because, of course, some stars are brighter in the blue than they are in the red. Some stars are brighter in the red than they are in the blue. What does the average work out to be? It's either surprising or unsurprising that the answer is beige. The universe is beige. And I hope that color comes out as slightly different from the background white there. Nobody wanted to call the universe beige or magnolia, so they called it either cosmic latte or my favorite, skyvery. I quite like skyvery. That's quite a nice way of describing. So it's perhaps not surprising that, yes, of all the stars that we can see, there will be blue stars, there will be red stars, no green stars. And when you merge those all together and add them all up, not surprisingly, you get something which is fairly insipid color like that. So let's stick with skyvery. It sounds much better than beige. So we've dealt with the colors of stars. We've said stars are black body objects. They emit light with a particular range of wavelengths. According to the laws of physics, you can't get anything else. At a given temperature, you will get a particular distribution of wavelengths. Hence, we saw red giants emit mainly in the infrared. Our sun emits mainly in the visible. Very hot stars emit mainly in the ultraviolet. But the colors of nebulae are rather different. They don't obey the same rules. So the question is, why not? So if we think of stars and galaxies, so let's just simplify this and say galaxies are just collections of stars. Actually, they're collections of stars and gas and dust and maybe a few other bits and pieces. But let's assume that when we're talking about galaxies, we're just talking about the collection of all the stars within those galaxies. If we were to look at the spectrum, we would find it's a continuous range of colors. I'm not trying to imply here that when you look at a galaxy, you would necessarily see this spectrum. Some stars, of course, will be brighter in the red than they are in the blue. Some stars will be brighter in the blue than they are in the red. The point is, all colors exist in the spectrum of either individual stars or certainly when we're collecting stars together in galaxies. But if we were to look at the spectrum of a nebula, if we were to put that through a spectroscope and ask what does the spectrum look like, we get something that looks very different. So for instance, if we took a large cloud of hydrogen, this happens to be the Rosette Nebula, but it doesn't really matter. If we take a large cloud of hydrogen, we will typically see this for the colors when we look at the spectrum, either by putting it through a prism or putting it through a diffraction grating. Any spectroscopic technique will end up with a spectrum that looks like that. It's called an emission spectrum. Sometimes it's called a line spectrum because the spectrum consists of a number of lines because light is being emitted at very specific wavelengths. In the case of hydrogen, there appears to be a very obvious emission at the red end of the spectrum, and there appears to be one in the blue end of the spectrum as well. There are, in fact, lots of emission lines, but we're just looking at the visible part of the spectrum here. So there is a fundamental difference between the colors of stars and the colors of nebulae because their spectra look fundamentally different. So what is responsible for the difference there? Let's remind ourselves what atoms are. Well, that's not an atom. It's not a very good diagram, but it will do. It's got a nucleus. Every atom has got a nucleus and a whole load of electrons buzzing around the nucleus like bees. But one of the reasons that this is not a very good diagram for us thinking about how atoms actually work and what's going on inside an atom is that this gives us the impression that all the electrons are doing the same thing. They're all at the same distance from the nucleus. They're all going around the nucleus. Okay, some of them are going in different orbits, but all the orbits look the same shape, the same size. All of those electrons are the same distance from the nucleus. And that's why this diagram is fundamentally misleading when we're talking about atoms. Again, I'm not going to talk and turn this into a physics lecture, but a slightly better diagram of how we should be thinking about atoms is this one, in which it's beginning to look suspiciously a little bit like a solar system. We must put that out of our heads, but at least in this particular diagram, we can see that different electrons are different distances from the nucleus. And what's absolutely important in terms of atomic physics, as far as we're concerned, is that different electrons at different distances from the nucleus have different amounts of energy. And I'll say why that's important in a moment. So we know that atoms are not like a solar system. They're not flat. Atoms are usually a little more spherical. So we actually should be talking about a shell of electrons around a nucleus rather than something that looks a bit like an orbit of Mercury or Venus or Earth, etc. So we really ought to be talking about nested shells, which have been cut in half here so you can get the idea. But whenever you have a three-dimensional model, it's a little more difficult to draw, so we're going to stick for the time being with this idea. Of course, it's different to a solar system because in a solar system, Mercury orbits at a given distance and Venus at a different distance and Earth at a different distance. Here it appears that 2 electrons are at the same distance and then 8 electrons are in the same orbit, etc. Again, we don't have to worry about what are the rules that say how many electrons are at a given distance from the nucleus. There are a set of rules. It's called quantum theory or quantum mechanics that tells us how this all works. But the important thing is that every atom has a particular pattern, a particular fingerprint, a particular configuration, if you like, of how those electrons are arranged and moving around within the atom. And one of the fundamentals of our understanding about how electrons move from one energy level to another, different distances from the nucleus, is that if an electron loses energy, then light is emitted and carries away that difference in energy. And if light impinges on an atom, it can raise the electron energy. And the amount of energy that an electron gains or loses, that's the amount of energy that the photon of light either comes in with or goes out with. And the important concept that we need to get hold of is the fact that there is an energy scale when we look at photons of light with all of these different wavelengths. Remember, the blue end of the spectrum is at 400 nanometers. The longer wavelengths are the red end of the spectrum. So wavelength is going from short on the right to long on the left, but the energy scale is the other way around. So red photons have a lower energy than blue photons. So in other words, there's a reciprocal relationship between energy and wavelength. It doesn't matter so much as the fact that there is a one-to-one relationship. A photon of a particular color has a particular wavelength and it has a particular energy. So if an atom has got a particular arrangement of atoms, a particular configuration, then there will be certain jumps that can take place between different energy levels of that atom, and a particular energy will correspond to a particular wavelength, which will correspond to a particular color. So if we have an atom, this is not necessarily hydrogen because there's far too many electrons in this particular atom, but every atom has a particular arrangement of electrons, and because of the jumps between these different energy levels, if an atom has a particular arrangement of electron levels, that means there are particular energies of light that can be absorbed or emitted by this atom. And remember, particular energies corresponds to particular wavelengths. So that's why we see for any given atom, for instance, an atom of hydrogen, the electron in hydrogen can be in different energy states, and whenever the energy of the electron changes, a photon of light is either absorbed or emitted and will be a particular color because hydrogen has electrons of a particular set of energy levels. We're not going to describe how they're calculated. When we describe this in a physics degree, it takes a few years to understand why particular atoms have particular energy levels. All we have to worry about is the fact that they are there and we can calculate them. So we know that hydrogen produces a very bright line in the red end of the spectrum and another one in the blue end of the spectrum. If hydrogen happens to be not emitting light but absorbing light from a more distant object, for instance, if a hydrogen cloud has got a star behind it and that star is shining lots of wavelengths through the cloud will absorb, again, exactly those same wavelengths. It'll absorb a line in the red and it'll absorb a line in the blue. So the difference between emission and absorption is just literally, are photons being absorbed by the atom or are photons being emitted by the atom? The important thing is that the pattern of where those lines exist is a function of the atom. It is a characteristic, it is a signature of that particular atom. To the point that in spectroscopy, if we wanted to look at a particular star and see which particular lines existed, we can map one-to-one. If a line exists at that wavelength, that corresponds to this energy, that corresponds to this atom. So we can associate colors in the spectrum with particular atomic species. That is effectively the power of spectroscopy. If we were to look, for instance, at where those colors exist, if we look at a nebula, it might be shining because this nebula has got hydrogen atoms or oxygen atoms, and hydrogen will give an emission, as we've just seen, at the red end of the spectrum and the blue end of the spectrum. Oxygen gives one sort of in the middle of the spectrum at about 500 nanometers or so. So if we've got a nebula such as we showed before, the Rosette Nebula, which contains hydrogen and oxygen, or the Veil Nebula, which contains a lot of hydrogen and oxygen, we will find those three colors show up. And essentially, nothing else. No other colors in here, if we're talking about a nebula, not a star. And if it's only made up of hydrogen and oxygen, we will see essentially just those three colors. But we still haven't answered the fundamental question, because we can say, okay, if it's made of atoms and those atoms have particular energy levels, then we get particular photons of particular colors, and so we get those particular lines. But we've said in the previous section, if we have stars of different temperatures, we get all colors. Well, why the difference? Because in this case, a nebula might be made of hydrogen, helium, oxygen, carbon. And what are stars made of? Well, hydrogen, helium, oxygen, carbon. They're made of the same things. So if nebulae and stars are actually made of the same stuff, why in one case do we get something that's characteristic of atoms, and in the other case we get something that's characteristic of what would happen if we heated up a bar of metal and saw how its colors changed and how the wavelengths of light changed as a function of temperature? So what is it that is fundamentally responsible for those differences? The difference is relatively straightforward. It is only the density of the material we're looking at. How many atoms do we have that are responsible for producing these colors? If the atoms are well separated, one atom doesn't really know about the other atoms. If you separate the atoms by more than a few microns, well, you might as well separate them by light years. One atom is not aware of the existence of another atom unless they are relatively close together. So if the atoms are well separated, they will all behave independently as little sources of light which give you the characteristic light of an individual atom. But if you take these atoms and put them all close to each other so they're always bumping into each other, then the spectra of all the individual atoms get smeared out until you end up with a continuous spectrum. So if you like, that is the fundamental emission from atoms, but the result we get from stars and anything else we heat up is simply because we've got so many atoms in there, they are bumping into each other and literally changing the energy at which we see these lines to the point where they get smeared out over the entire spectrum. So to understand what we would expect to get from any given object, all we really have to do is to work out the density. And the density varies by an enormous amount for different objects in the universe. So let's just consider, how many atoms are there in something the size of a sugar lump? How many atoms are there in 1 cubic centimeter? If we take a lump of metal, there are approximately 100,000 million atoms in a lump of metal. They are really close to each other. Atoms are separated by distances which are barely any bigger than an atom, basically. One way of thinking of it is the atoms are touching. Okay, that's not quite right in quantum mechanics, but it's close enough. The atoms are right next door to each other. Even if we go to the Earth's atmosphere, you might not think that the molecules and atoms in the air in this room are particularly close to each other. We find it very easy to move through the air, so you might think that the atoms are a long way apart. Well, they are a long way apart compared to a piece of metal, but there are still, 10 million of them in a cubic centimeter. So although, yes, air is much lower density than metal, it's still an absolutely huge number of atoms in 1 cubic centimeter. So they're still bumping into each other all the time. If we go to the surface of the sun, the photosphere of the sun, you may be surprised to know that it is substantially less dense than the air in this room. You might think of the sun as being a very dense object. Well, it is at the core, but the outer part of the sun, the surface of the sun, is actually significantly less dense than the Earth's atmosphere. The number of atoms of hydrogen and other elements per cubic centimeter is relatively low. Still a huge number. They're still bumping into each other, which is why the surface of the sun is a continuous spectrum. If we go out to the sun's atmosphere, the corona, although it's a lot hotter, it's a very much lower density medium, a very low density plasma. And you can see we're dropping substantially now. We now only have 10 million atoms per cubic centimeter in the solar corona. You might think that 10 million is a lot, but in terms of atomic speak, that's a tiny number. If we were to go further and ask in an interstellar nebula, if we were to ask what's going on in the Trifid Nebula or in the Rosette Nebula, how many atoms do we have in there? The atomic density is really tiny. If we were to go to the interstellar medium and start talking about not a cloud of hydrogen, but effectively an area of nothing in particular between us and Alpha Centauri, if we go to the interstellar medium, we're down to the point where we've barely got 1 atom per cubic centimeter. So quite definitely, any atoms in these sorts of either nebula or interstellar medium are not aware of the other atoms. If we take it to the extreme and say what's happening in the intergalactic medium, well, I don't mean zero in the strict sense that there aren't any atoms out there. There are definitely atoms floating around in between galaxies. But instead of talking about how many there are in a cubic centimeter, we really ought to be talking about how many there are in a cubic meter. And there still might be 1 or 2 or 10 in a cubic meter. So bearing in mind how far it is from here to Andromeda, that's an awful lot of cubic meters. So there's still an awful lot of atoms between us and the next galaxy. But you can see the numbers change dramatically, and that determines whether the spectra of these objects is going to be essentially atomic-like, or whether the atoms are so close to each other they smear the spectrum out into a continuous spectrum. And hence, with these really big numbers at the top, we would expect these objects to give us effectively continuous spectra, all the way down to the surface of the sun. But for everything else, from the solar corona through nebulae and the interstellar medium, the atoms are so far apart, they effectively give atomic spectra. So even if they're made of the same stuff, when we look with a spectrum, with a diffraction grating, with a prism, once we spread the colors out, we will see fundamental differences in the spectra of these objects purely based on how dense these objects are. I'm not specifying exactly how dense it is before it transitions, as it were, from one to the other, but the sun is definitely dense enough to be a continuous spectrum, and the corona is definitely dilute enough to be effectively an atomic spectrum. Let's just think about going beyond color before we finish. If we think about Hubble Space Telescope images, this is a question that's often raised by people that don't understand how imaging is actually carried out. For instance, if we have a look at the pillars of creation, the question you often get is this a real picture? This is a color image, but is it a real color image? Is that what it would look like if we were, as it were, standing in front of it? Now, we know our color vision is not good enough to see faint objects, but if you just put that to one side and imagine that the pillars of creation were much brighter than they actually are, would that be the colors that we actually see? Quite definitely not, because most objects that are out there are made of hydrogen. And so this is a nebula, it's made of hydrogen and other things. But if it's mainly made of hydrogen and hydrogen glows mainly in the red, then essentially every nebula that you would look at is red. And for those that have tried astrophotography, it's no surprise that when you take images of nebula, if you don't use filters, generally you always get a red result. But to take images of nebula and always get the same colors is a bit boring. If you want to understand the structure of these things, it's more useful not to take a color image where everything comes out red because it's dominated by hydrogen. It's more useful to say, I'm not going to take a red, green, blue image by filtering out what's coming in the red end of the spectrum, what's coming in the middle of the spectrum in the green range, what's coming in the blue end of the spectrum. I'm not going to simply use filters just like a normal color camera that separates red, green, and blue, because that would be dominated by the hydrogen in the red. What we're going to do in the case of the Hubble Space Telescope and many other imaging techniques, is to say we're going to take an image in which we're only going to let through the light of one element. And we're going to take another image where we're only going to let through the light of a second element. And then we can do the same with the third element. There's no reason why those three elements that you're imaging necessarily fall into the red end of the spectrum, the green bit of the spectrum, and the blue bit of the spectrum. The conventional way of imaging with the Hubble is the so-called Hubble palette. This is just a convention. There's no reason why you have to use this, but it's a convention that says a lot of nebula will contain various elements. They will contain hydrogen, oxygen, and perhaps surprisingly, quite a bit of sulfur as well. So sulfur, hydrogen, and oxygen, the wavelengths of those particular elements, the wavelengths that are dominant for those particular elements. Hydrogen comes out in the red, but so does sulfur as it happens. Oxygen tends to come out in this sort of teal part of the spectrum. So this particular image of the pillars of creation, the image that we declare as red was actually taken from sulfur. But of course, it's almost the same color as hydrogen, but it's a different wavelength. So if you use a different filter and take images at that wavelength, you're picking out the hydrogen. And then you can basically say, I want that to be green when I combine these images. I'm going to combine them as red, green, and blue to make a color image. You can do what you like. If you take three wavelengths, you can combine them together how you like. The convention for the Hubble palette is that if you want red, green, and blue, which remember is long wavelength going down to short wavelength, you keep the ordering the same. You don't have to do that. That's just a convention that astrophotographers do. So the fact that these two look very different is not simply because they are, you look at that and you say, well, they are very similar wavelengths. Why should they look different? Because they are imaging different different elements. The fact that they're only a few nanometers apart is neither here nor there. You are taking a very narrow band of light and picking out what is the hydrogen doing. You're taking a narrow band of light and saying, what is the sulfur doing? And then when you've done that for sulfur and hydrogen and oxygen, you get three images, and arbitrarily you say, I'm going to make that one red, make that one green, make that one blue, add them together, and hey presto, I've got a color image. And because I am imaging the sulfur and imaging the hydrogen and imaging the oxygen, in this particular case, you do not get something that's dominated by the red, because you've got a fair amount of green, remember that's hydrogen, and you've got a fair amount of blue, that's the oxygen. So you've got a reasonable balance of the red and the green and the blue. It is not the actual color of the pillars of creation. It's an image of the pillars of creation in which the colors that you are able to perceive in the final image allows you to extract a lot of structural information because you're seeing a lot of what elements are there. The fact that you don't really care which color corresponds to which element, again, doesn't really matter. It has been chosen by the people who took the image to try and reveal as much structural information as possible in the final images. And it really doesn't matter, other than for aesthetics, the fact that that is not the actual image in terms of the actual true colors. Any amateurs that try taking an image of the pillars of creation, as I say, will tend to get something that looks very pink, unless they do the same thing. Use a monochrome camera and use filters that allow you to pick out the sulfur, the hydrogen, the oxygen, combine them together, and then you too can generate images with the so-called Hubble palette. Some amateur astrophotographers like to do that because either they like to produce images which are similar to the Hubble Space Telescope, or because genuinely it does show you more detail than just taking a color image in the conventional red, green, blue sense. So, do Hubble Space Telescope images represent true colors? Well, in some cases, yes, if they're taking pictures of galaxies. Remember, galaxies have wavelengths that cover the entire spectrum. It's nebulae that tend to have particular wavelengths corresponding particular atoms. When you're dealing with low density objects like nebulae, it's interesting to do this. If you're talking about stars, which are effectively high density objects, all colors are present, there's really no point in doing this. So when Hubble takes a picture of a galaxy, it won't necessarily pick out this. It might well be a true color image of the galaxy. So does Hubble Space Telescope images represent true color? Strictly speaking, it depends on the object it's imaging. In this case, no. In the case of galaxies, perhaps. And remember that the wavelengths we're talking about, from 400 to 700 nanometers here, that of course is only a tiny fraction of the full electromagnetic spectrum. So we're familiar with what's going on here, and we sort of know about ultraviolet and infrared. It's relatively easy to build telescopes that look a little way into the ultraviolet. The Hubble Space Telescope does that. And we know about telescopes that look in the infrared, such as the James Webb Space Telescope. But if you're working entirely in the infrared, then of course the images cannot be true color images. There's no such thing as a true color image if you're working in an area outside of the visible spectrum. You can take an image at this infrared wavelength, and you can take an image at that infrared wavelength, and you can combine them together by artificially coloring that one red and that one green and that one blue, but it's entirely arbitrary. Usually, again, by convention, if you're taking images at a number of wavelengths, you normally stick to the same order. So the colors which are assigned when you finally produce your image are in the same order as the particular filters that you've used. The filter wheel of the James Webb Space Telescope is incredibly complex. It has a shed load of filters that it can use, not just red, green, and blue, but basically any element that could conceivably be imaged. Well, there's a filter that will let through light for that particular element. And so by combining images taken with lots of different filters, you can get all of the information you want out of a James Webb Space Telescope image. There are lots of different filters. Some let through quite a wide range of wavelengths. I know you can't really see the detail here. Some filters let through a broad range of wavelengths. Some filters let through only a tiny range of wavelengths. It really depends on what science you want to get out of those images. Are you trying to identify particular features, particular elements, or are you looking to get the gist of just what the overall structure of this object is? So let me summarize the take-home messages from this talk this evening. The number one is probably your eye is not a camera. There are so many differences between the human eye and the camera that's either in your phone or in the camera that you use for astrophotography. The eye does not simply detect color intensity and send that information to the brain to allow your brain to make a color image. There's far more processing that goes on in the neurons in the back of your eye that sometimes leads to your brain getting fooled into you thinking that you're looking at something that you're not actually looking at. The colors of stars are dominated by this physical phenomena called blackbody radiation, where when something is hot, it emits light of particular wavelengths according to a particular curve, the so-called blackbody radiation curve. And so red giants emit in the infrared, the sun emits in the visible mainly, and hot stars emit in the ultraviolet. And so the colors of stars are either red or whitish, sometimes yellowish, whitish, or blue-white, but there are no green stars. The colors of nebula are completely different because now we're talking about atoms that are well separated. So individual atoms produce a spectrum which is identifiable as a fingerprint of that particular atom. And therefore, if we look at the emission from nebulae, we can see what they are actually made of. Scientifically, an absolutely enormously valuable process to actually tell us not only that looks interesting, but by looking at the spectra, we can say what atoms actually make up that nebula. And I haven't had time, but there's a bit of information in the handout there, because generally spectroscopy can be used not only to look at the colors of stars, we can also use spectroscopy, for instance, to measure redshift to distant galaxies and things like that as well. But I really wanted to spend time this evening just making sure everybody understood the colors of the cosmos in terms of why the colors of the stars are different from the colors of nebulae. Thank you all very much.